Hi, I am Jancee Tapayan, and today we will discuss about the central dogma process, starting from replication, transcription, and translation. Central dogma is the process by which the instructions in DNA are converted into a function product. It was first proposed in 1958 by Francis Crick, discoverer of the structure of DNA. The central dogma of molecular biology explains the flow of genetic information from DNA to RNA to make a functional product, a protein. DNA, or deoxyribonucleic acid, the double-stranded helix whose backbone is made up of deoxyribose sugar and phosphate. The two backbones are connected by four different nucleotides: adenine, thymine, guanine, and cytosine. In DNA, adenine pairs only with thymine, while cytosine pairs only with guanine. Replication. In replication, the DNA, which is double helix, is uncoiled and the strands are replicated to form two identical DNAs. Double helix is broken down into single strand by helices. The helicase is an enzyme used during replication to break the hydrogen bonds between the complementary base pairs into two separate strands. After helicase divides the DNA, a different types of enzyme called DNA polymerase comes in and attach itself on a strand. It catalyzes two nucleotides to bond together so it produces a new strand. It is a strand that is made by replicating old original strand, and the parent strand refers to pre-existing single strand that is being replicated. This replication described semi-conservative because half of pre-existing DNA is always will remain the same. Transcription is a process where a section of DNA is replicated to produce RNA. A type of enzyme called RNA polymerase moves along the DNA to unzip it into two strands. RNA polymerase connects to one strand of DNA using it as a template. Template strand is where mRNA is to be made. After it's complete, it comes off and leaves. In RNA, guanine is paired with cytosine like in the DNA. Thymine in the DNA pairs with adenine, but in RNA, adenine pairs with uracil. After mRNA finishes replicating, it detaches itself and moves on the next process. This is the end of transcription and the beginning of translation. Translation. In this step, the nucleotides are converted into amino acid. Thereby, proteins are synthesized. These occur in the ribosomes. A tRNA with specific set of anticodons and amino acid comes and binds with the codons of the mRNA. Codon is made up of three adjacent nitrogenous bases. A second tRNA brings a specific amino acid next to the first tRNA. An enzyme connects to two amino acids using condensation, and the first tRNA will break off its amino acid and exit. The second will have more room for the next one, and so on until it will stop. Now you have a protein.